Hello students, I am Dr. Ashwini Karve and I would be discussing pharmacodynamics or in simple language, drug action in this module. Let us see what are the specific learning objectives of this session. At the end of the session, first year homeopathy students should be able to define drug and pharmacodynamics, explain the mechanism of drug action, enumerate factors affecting drug action, explain the terms agonist, antagonist, efficacy and potency, define adverse drug reaction or ADR, understand difference between side effects and ADR, and enumerate different types of ADRs. Let's start by defining a drug. So drug is defined as any substance or product that is used or is intended to be used and to do what? To modify or explore psychological systems and pathological states for the benefit of the recipient. It can also modify the physiological systems. So with this definition in mind, let us now start the session by understanding the meaning of the term pharmacodynamics. As you can see, in any therapy, there are two systems involved: drugs or medicines and human or patient. Naturally, both would have effect on each other. What the human does to the drug is called as pharmacokinetics. And what we are interested today's session is what the drug does to the human body, and that is called as pharmacodynamics. In other words, pharmacodynamics is the study of the drug effects or action. Let us now understand how the drugs act on the body. That is, what are their mechanism of actions? There are three main mechanisms: physical, chemical, and by acting on a target. Further, the target is of four different types. That is, an enzyme, ion channel, transporter, or receptor. Let us see each one in little detail by taking an example so it becomes easy to understand. Physical means the drugs act by its sheer presence in the body. Let's take an example of bulk laxatives. These are used to treat constipation. That is, the patients who have difficulty in passing the stools would be given or advised bulk laxatives. So when these bulk laxatives reach large intestine, they absorb water, and along with the formed stools, they form a bulk. Now this bulk then stimulates the peristaltic movement of large intestine, and as a result, results into evacuation of stool. So here, if you can see, the drug has merely acted by its physical presence in the body. So that was physical way or physical mechanism of action of drug. The second way the drug can act is by chemical reaction. Here, the drug reacts with the substance in the body chemically and produces the action. Classic example I would like to quote here are antacids. Now antacids are drugs that are used when patient complains of acidity or peptic ulcer. So as shown in the picture, sodium bicarbonate is a type of antacid. When taken in the stomach, it reacts with the acid and neutralizes the same by producing water and carbon dioxide. Thus, it results in reduction of acidity. It neutralizes the acid in the stomach. So in this case, the drug has acted chemically in the body. In the next few slides, I would be talking about action of the drug by attaching to a particular target. Almost all biological reactions require enzyme as a catalyst. Thus, enzyme becomes an important target for drugs. Let us see how it works. Every enzyme has an active site where the substrate, that is a natural substance present in the body, will attach. And this will lead to a biological reaction and many a times results into degradation of that substrate. But when there is a presence of a drug that binds to the same active site of the enzyme, the substrate now cannot bind and the reaction does not happen. Let me explain using an example. Cholinesterase is an enzyme. Acetylcholine, a neurotransmitter involved in many important functions, is its substrate. When acetylcholine attaches to cholinesterase, cholinesterase enzyme degrades acetylcholine. Now there is a drug called physostigmine. This physostigmine has the ability to bind to the active site of the enzyme cholinesterase, and thereby it blocks the binding of acetylcholine to this site. So now cholinesterase enzyme is unable to degrade acetylcholine. As a result, concentration of acetylcholine increases and then it continues to produce the required action. So in short, physostigmine here prevents degradation of acetylcholine by attaching to cholinesterase as an enzyme for its target. Second important target is ion channel. As you all know, ion channels are used by different ions in the body to move in and out of the cells. By this movement, the normal intracellular and extracellular ionic composition is maintained. So, drugs used these ion channels as their targets. By attaching to ion channels, drug would either increase or decrease the ionic movement across the channel, and that would bring about the drug action. Different drugs would bind to different ion channels and produce different actions. Let's take an example of sodium channels present in the nerves. Movement of sodium is required for propagation or for the traveling of nerve impulse. There is a drug called lignocaine which is a local anesthetic. That means it produces loss of sensation over a local designated area. Now this drug binds to sodium channels and blocks the entry of sodium inside the cell. This results into blocking of propagation of nerve impulse and ultimately resulting into loss of sensation over an area which is supplied by that nerve. So here, lignocaine acts as a local anesthetic by acting on an ionic channel as its target. Many substances use specific transporters for their transport. Drugs can bind to such transporters and produce actions by interrupting the normal process. Let me explain the same using an example. This is a neuron where 5-HT or serotonin is released. The action of serotonin is terminated by movement of serotonin back inside the neuron, and for which it uses SERT, which is called as serotonin transporter. There is a class of drug called SSRIs or selective serotonin reuptake inhibitors. Now these drugs bind to the transporter, serotonin transporter, and prevent entry of serotonin in the neuron. Thus, serotonin concentration in the synapse increases. So does the action of the serotonin. This serotonin is required for uplifting of the mood of the patient. Thus, SSRIs, the drugs, by increasing its concentration, that is increasing the concentration of serotonin, result into uplifting of the mood of the patient. And therefore, these drugs are used in the treatment of depression. So this is how the drug acts, by acting on a transporter as a target. The most important target and the one which maximum drugs use is receptor. What is a receptor? Receptor is a macromolecule present either on the cell membrane or inside the cell. In our body, there are multiple types of receptors. Each one will have a variety of body substances attaching to it and producing different actions. The diagram which is projected is of a receptor present on the cell membrane. Let us now see a few terminologies which are commonly used while explaining the drug action via receptors. The first one is ligand. It originates from a Latin word meaning to bind. So ligand is any molecule which attaches selectively to a receptor. This molecule could be an internal substance or it could be a drug. The second important term is an agonist. An agonist is a substance that binds to a specific receptor, activates it and produces effect, while antagonist is a substance that binds to a specific receptor and blocks the site, so that now agonist cannot bind there and produce action. In short, antagonist prevents the action of an agonist on a receptor, but on its own, antagonists have no effect in the body. Remember that both agonist and antagonist are ligands, that is, they bind to the receptors. Thus, a drug acting via receptor could be an agonist or an antagonist. Let's take one example to explain this. This is a receptor site and its physiological substrate. For example, the receptors are beta 2 that are present on the bronchial smooth muscles, and they bring about bronchodilation. The natural substrate for beta 2 receptors is adrenaline. Now the agonist will bind to the receptors and bring about enhanced activity. For example, a drug named salbutamol, which is an agonist of beta 2 receptors. Salbutamol binds and causes bronchodilation, and therefore it is used in the treatment of bronchial asthma. Now there is another drug called propranolol, which binds to the same beta 2 receptors, but propranolol is an antagonist, and hence it blocks the action of salbutamol, thus prevents bronchodilation. Thus, salbutamol is beta 2 receptor agonist and propranolol is beta 2 receptor antagonist. Now we know what are ligand, agonist and antagonist. Let us now understand two more terms, affinity and intrinsic activity. Affinity means ability of a substance to bind to the receptor, and intrinsic activity means ability of a substance to produce effect. So applying these two concepts, one can say that an agonist has both affinity and intrinsic activity because it binds to the receptor, but in addition it also produces action. So it has both affinity and intrinsic activity. On the other hand, an antagonist. Antagonist has affinity because it is able to bind to the receptor, but it has no intrinsic activity because it only binds but does not produce any action. So these are the terms affinity and intrinsic activity. Now let us proceed with the module. As I was explaining to you about the receptors, I said that a drug could be an agonist or an antagonist. Agonist is the drug which has both affinity and intrinsic activity, while antagonist is the drug which has affinity but no intrinsic activity. So then I concluded saying a drug can act as an agonist or an antagonist. Let's start talking about a little bit detail about the receptors. There are 4 types of receptors depending on the structure of the receptor and the way it produces the effect at a molecular level. They are receptors with intrinsic ion channel, G protein coupled receptors, enzyme linked receptors, and receptors regulating gene expressions. The first one in the picture is intrinsic ion channel receptor. As the name suggests, these receptors have intrinsic ion channels in them. The second type is G protein coupled receptor. The activation of these receptors would result into activation of gene protein, and there would be series of events which ultimately would lead to action. The third type of receptor is enzyme linked receptor. Here the receptor has enzyme as its subunit. So an enzyme is attached to the receptor. The activation of receptor and then the activation of enzyme would result into an action. And the last or the fourth type of receptor is slightly different. It is not present on the cell wall as the earlier receptor types, but this receptor is present inside the cell. Now what happens? The drug enters the cell, attaches to the receptor, and now this complex, that is the drug and the receptor, is then transported inside the nucleus. The effect is produced by altering the gene expression. So these are the 4 types of receptors. If you see the response time after the receptor is being activated by a drug, then it is quick or fast for the ion channel receptor, while longest time is taken for the receptor regulating the gene expression. Thus, the onset of action of drugs acting via ion channels is quick as compared to the others, while the drugs acting via receptors regulating gene expression would be much more. Next, let me explain the 2 terms which are commonly used while talking about drug action. The first one is potency of the drug. It involves the amount of drug required to produce a particular action. Lesser the amount required to produce the action, more potent is the drug. Let's take one arbitrary example. Say 20 mg of drug A produces a particular action. And now to produce the same action, 100 mg of drug B is needed. So if I have to say between drug A and drug B, I can comment or I can say that drug A is more potent than drug B, because drug A only in 20 mg produces the action, while to produce the same action, drug B requires more concentration. So then drug A becomes more potent than drug B. The next term is drug efficacy. Now what is it? It is the maximum response that can be elicited by a drug. More is the response, more efficacious is the drug. For example, a response produced by drug A is more than response produced by drug B. And here when I say response, it's the same response. then we can say or we can conclude that drug A is more efficacious than drug B. So these were the two terms, potency and efficacy. Let's talk about certain different aspect. We all know that every individual or patient is different, and hence the action of the drug may be affected by various factors. Thus, the dose of the drug or even sometimes the choice of the drug would be adjusted based on certain factors. So that means let's see some of the factors which affect the drug action. The first factor is body size of the patient. The usual dose of the drug is for the individual of the average build. Now naturally, for obese patients and patients who are very thin and even patients who are children, the dose would be different. And then the dose is calculated based on their weight as follows. There is a formula which says the dose of that patient would be body weight of the patient in kilogram upon 70 into the average adult dose. That was about body size. The two extremes of the age, that is children and elderly, that is old patients, would require alteration of the dose. The reason? Children body system are not as developed as adult. They are developing yet. While in elderly, their body functions are slowly declining. So then both these age groups, one would have to alter the dose as compared to that of normal adult. We all know that physiological changes happen during pregnancy. Now these changes will also affect the action of the drug. So the drug dose needs to be adjusted in a pregnant lady. Also, one has to remember that the drug taken by the mother would definitely have an effect on the growing baby. So it becomes important to select a drug which is safe for the growing baby. The next factor is the genetic factor. You have to remember that the genetic makeup also plays an important role in the action of drug. In fact, there is a separate branch called pharmacogenetics, and this branch deals with effect of genes on drug action. Let's take one example. Some individuals are deficient in producing a certain enzyme. Now, one such enzyme is G6PD. There is a drug called primaquine. Now, this is the drug which is used in the treatment of malaria. Suppose the patient is deficient in the enzyme G6PD and that patient receives primaquine, then there would be bleeding in such individual, and which could be fatal. Therefore, it's a routine practice to do a blood test and find out whether the individual is G6PD deficient or not before starting the primaquine treatment. So mind you, genetic makeup of the patient can also have an effect on drug action. We know that the drug can be given by different routes to a patient, and that could also have an effect on the drug action. For example, usually a drug is given by injection would act faster than the same drug given orally. Also, it needs reduced dose of the drug to produce the action when given by injection compared to that of oral route. So the route of administration of the drug can also have effect on the activity or action of the drug. The psychology of the patient also plays a role in drug action. It is evident when sometimes placebo is used in the therapy. Now, what is this placebo? Placebo is an inert substance. It has no activity on its own. But sometimes physician feels that the patient does not require any drug therapy. But on the other hand, patient refuses to accept the same. And patient in fact insists that he has to be prescribed some drug. So in this situation, what the physician would do? Physician would prescribe placebo. Of course, physician does not tell the patient that he has prescribed him placebo. So then what happens? It works on the psychology of the patient. Patient feels that yes, I am being treated by the physician by this particular drug, and that actually helps to cure the so-called disease in that patient. So psychology, mind you, also plays an important role in the action of the drug. We know our body has an important organs in the form of liver and kidney. Now these organs are involved in metabolism and excretion of the drug. So if a patient is suffering from diseases of these organs, suppose patient has hepatic failure or patient suffers from renal insufficiency, then definitely the physician would have to alter the dose before giving it to that patient. Nowadays, you must have realized that one patient takes more than one drug at a time. So then if those drugs are going to act on the same target in the body, then both the drugs would interact with each other, and therefore in such situation, the dose of the drug would definitely need to be altered. So then students, till now we have discussed the first 4 objectives of our SLOs. Let's talk about the other 3. We all know that many a times a drug would produce an action which is not wanted. And in fact, it may cause harm to our body. Such actions are called as adverse drug reactions or ADRs. What is the definition of ADR? It is any noxious change that is suspected to be due to a drug that occurs at doses normally used in the man, that requires treatment and or decrease in the dose, or indicates caution in the future use of the same drug. There are many ways one can classify ADRs. I have taken these 2 common types which are easy to understand. Type A, where A stands for augmented or increased, and type B, where B stands for bizarre, that is without any explanation. So type A reaction is due to the pharmacological property of that drug, while type B is due to peculiarity of the patient. So that means type A is drug dependent, while type B is patient dependent. Type A is dose dependent. More the dose, more would be the adverse drug reaction, and it is commonly seen. While on the other hand, type B is independent of the dose. Even the small dose can give rise to adverse drug reaction in a particular patient. But type B reactions are relatively rare. Type A reactions are preventable, and also they can be reversed, because once you stop the drug, the adverse effect can be reversed. On the other hand, type B are more serious as compared to type A, and most of the times type B reactions, once it happens, requires withdrawal of the drug. Let's take again an example. Atropine, which is an anticholinergic drug. Now, as a part of its pharmacological action, atropine reduces or decreases the secretion of various glands of the body as a part of its pharmacological property. And thus, patient would complain of dry mouth, because now what has happened is the salivary gland secretion has gone down. So then that would be an adverse drug reaction. This reaction is dose dependent, and most of the patient would complain of dry mouth with atropine. While on the other hand, penicillin, which is an antibiotic. Now penicillin can produce anaphylaxis. Now anaphylaxis is a very, very severe type of allergic reaction. This reaction to penicillin is not because of extension of its pharmacological action. Some patient would suffer from it, some won't. So it's very, very patient specific. So then allergic reactions would definitely fall into type B adverse drug reaction. So atropine resulting into the dry mouth is type A reaction, while anaphylaxis due to penicillin or allergic reactions due to penicillin would be classified as type B adverse drug reactions. Many a times we use the term side effect. For any untoward reaction produced by the drug, we said that this is a side effect of a drug. No doubt, side effect is an adverse drug reaction of the drug. But mind you, all adverse drug reactions need not be called as side effects. Now, why I am saying so? Side effect is a type A adverse drug reaction. That is, it is an extension of pharmacological effects of the drug. So, talking about the same earlier examples, I would say dry mouth produced by atropine is side effect of atropine. But anaphylaxis produced by penicillin cannot be called as side effect of penicillin. It is an adverse drug reaction of penicillin. Side effects most of the times is context or situation based. Now, what do I mean by that? Let's again take one example. There is a drug called codeine, and this codeine has two main pharmacological action. First, it suppresses cough, and second, it causes constipation. So when codeine is actually used to treat cough in a patient, that patient would complain of constipation, and that would be labeled as side effect of codeine. But in some other case, in some other patient, when the same codeine is used to treat diarrhea, now here constipation codeine produces would be actually the desired action of the drug and not labeled as side effect. Now this is what I mean by side effect, which is situation based or context based. So till now we've seen what is ADR and types of ADRs. But then the question would always come to your mind, how would one detect or find out adverse drug reaction of drug, especially when the drug is a new drug, it has newly come in the market? And how would the treating physician would be aware of the adverse effect of a drug? To get answers to this question, there is a program which is called as Pharmacovigilance. Now what is this? This program actually runs at the national level. So in India also we have Pharmacovigilance program, and also at international level. What is this program? It is a science or activities relating to what all it does: detection, assessment, understanding and prevention of adverse effects or any other drug related problem. So it's a continuous process. The data is collected by different physicians, and then it is analyzed and then decided if the ABC reaction is due to that drug or not, and then the information is shared by the other physicians, so they become aware of any new adverse drug reaction due to a particular drug. So then, students, to quickly summarize, in this module, we discussed, as I said earlier, about the drug and pharmacodynamics. We discussed in detail the mechanism of action of drug action. We also quoted certain examples so that the understanding of mechanism of different drug actions becomes easy. We quoted and explained certain terms such as ligand, agonist, antagonist, potency, efficacy. Then we also talked about the affinity and intrinsic activity of the drug. We also talked about certain factors which affect the drug action, namely body size or pregnancy, age of the patient, child or old age group patient, the route of administration, psychology of the patient, genetic factor of the patient, any important toxicities which are there or any important organ dysfunction. All these factors would definitely have the effect on the drug action, and we discussed it in detail. Then we also discussed about pharmacovigilance, which is a program in India as well as in international, which gathers and analyzes the data about adverse drug reactions. And there was a mention about the difference between side effect and adverse drug reaction, with two types of adverse drug reaction, that is type A, which is augmented or extension of pharmacological action, and type B, which is bizarre type of action. So these are the suggested readings. If you want to know the details about pharmacodynamics, you can go through any of these references and get to know much more knowledge about pharmacodynamics or drug action. Thank you so much.